So, today we will be discussing two topics that are very close to my heart, because much of our work is also going into these two questions. The first question is, it has to do with the implementation of quality control that I was mentioning the days before. That is, how we make sure that antigen receptors expressed on lymphocytes are selected against when they recognize self. And you probably know, of course, that this recognition of self is not an all or none, but a graded phenomenon. So we will be discussing this in a bit more detail, and then we will concern ourselves with the organ for one of the cell types, the T cells, that you by now must have realized I'm quite fond of. So the organ that regulates differentiation and selection of T cells, that is the thymus. And I'll give you some original experiments to tell you what one can possibly do and what we still cannot do in order to understand these mechanisms. And then in the second half of today we will be discussing the role of MHC in providing the molecular basis for eco-immunology. And I mentioned this from time to time, and I promised you that we will come to the second most important thing in human life, apart from money. And I will explain to you what kind of work we did along these lines to explain to you that MHC and the genetics behind MHC not only are important for immunology, but this also concerns the population structure, sexual selection, mate selection, and the success of populations in competition with pregnancy and ecological niches. So let's begin with the selection problem, and it is particularly virulent in mammals. And you can easily see why that is so, and I put this up here. Mammals have a particular problem when it comes to reproduction, because their young live in the mother's body for quite some time before they are released into the environment, as it were, whereas, for example, fish, they just have external fertilization. They are not concerned with this particular problem. Here we basically have a pre-programmed situation where there is an allograft in a mammal. So when the mother conceives, the fetus, by definition, because of MHC diversity that we already mentioned, is an allograft. So there is no one-to-one correspondence of MHC structure between the fetus and the mother. So that means that pregnancy is associated with the problem to modulate immune responses, otherwise the mother would reject this allograft, that is the fetus, and pregnancy would be terminated. So that is a major problem, and that is a problem that the immune system of mammals has to deal with, and we can now begin to understand what kind of modifications mammals have incorporated into their immune system to deal with this particular problem. In more general terms, you could say, because this is an allograft, you have to immunomodulate your immune system to prevent fetal loss. And at the first approximation, you might say, okay, this could be achieved by simply making the immune system of the mother anergic. So you could simply tell the immune system, as it were, stop, do anything, just wait until the baby is born, and then you come back to normal function. That, of course, is not the most fantastic strategy, because then during pregnancy the mother would be very susceptible to all possible parasite attacks. So there has to be a balance, and that balance, of course, is mirrored in the structure of the placenta, which brings the fetus close to the mother and the mother close to the fetus. This is where the action is, basically. So the placenta forms the barrier, and the placenta is a key element in this regulation or immunoregulation. However, we also know from clinical observations that females, women that suffer from autoimmune disease, that is for reasons that mostly are unknown, that the immune system of a female attacks her own body or particular types of the body. For example, the joints, so in rheumatoid arthritis, or the skin in systemic sclerosis. So when there is an autoimmune reaction going on, more often than none during pregnancy, the clinical symptoms are much less severe than before and after pregnancy. So this, I think, suggested from the very beginning, apparently, that there was some modulation of the immune system. Otherwise, these immune reactions would have continued unabated, but they don't. It's not true for all autoimmune disorders, I should say, but some of them are quite, well, this is certainly more frequent than this one. But in some frequent conditions, there is amelioration of the symptoms. So there is immunomodulation. And because there is immunomodulation, we must think of mechanism why that might be possible. And to understand this, and we still don't understand it actually, but to understand this, we have to go to the basics and understand how the system begins to develop. As you all know, in mammals there is an interesting biological problem in addition to the immune system, and that is the fetal-maternal conflict. You could even say there is a conflict between parental and between the two parental genomes, because the female carries the baby, and the parental genome, of course, wants to dominate the outcome of the pregnancy. So if, for example, parental genes dominate growth of the fetus, the female has to dampen the growth of the fetus, otherwise it would not be possible to deliver the baby, or the baby would consume too much energy from the mother, because of course there has to be some sort of balance. So there is always this sort of conflict. The fetus would like to grow as much as possible, and that is driven by paternal influence. The mother wants to restrict this, of course, but it has to allow growth, otherwise it would not work, the reproduction. But there is this competition between the two. And what I will not be discussing, but if you're interested in this, people think that genomic imprinting, that you might have heard about, has arisen to deal with this fetal-maternal or father-mother conflict during placental reproduction. Because the paternal genome drives growth, the maternal genome dampens it. And when you look at imprinted genes, those genes that have a differential expression depending on from which parent they come into the fetus, for example, IGF, insulin growth factor, that determines their activity. And this fetal-maternal conflict is not only represented in this imprinting and all these other conflicts, but of course it is being played out by the MHC. Now, what one has found very recently, you can see that here, this was sometime last year, a paper by Sasha Rudensky. What he and his group had done, they have looked at the effect of eliminating a particular genetic regulatory element in this FOXP3 gene that we have mentioned before, FOXP3 being a transcription factor that regulates the expression of regulatory T cells that can dampen immune responses. So there is one way of regulating immune response through these particular cell types. Belongs to the helper lineage, you might remember from yesterday. So these helper cells, or this special type of helper cells, can dampen immune responses. They are called suppressor cells. So they dampen immune responses, and when you manipulate the genome and you cannot build up these regulatory T cells, this is indicated by this red cross here, by manipulating the gene structure, the details don't really matter, but that results then in the abortion of this growing embryo, because the effector T cells that are marked here in blue will go unchecked by this regulatory loop. So the T cell activity here in the placenta is somewhat regulated, suppressed or dampened to a certain degree to allow this allograft to develop in the mother. If you take away this regulatory arm of the system, the fetus will not survive. So this is another example how the fetal-maternal conflict is being played out. It can be a special cell type that we mentioned before, and even outside of pregnancy, these regulatory T cells are very important. I discussed that yesterday when I was mentioning that these regulatory T cells are important in regulating intestinal microbiota, and their lack of those would then cause severe inflammatory bowel disease, a very severe autoimmune reaction, and these infants rarely survive. So there are ways at the molecular and cellular level to regulate these processes, but of course the T cells must be somehow also selected, or the repertoire must be selected. We've been discussing selection against self-reactivity for quite some time. So how is it possible, if we prepare the antigen receptor repertoire ahead of time before they see antigen, so how is it possible to purge this anticipatory repertoire of self-reactivity? And I mentioned some hours ago that, of course, when an embryo goes through development, some genes are active only during early development. They might be reactivated later during tumorigenesis, for example. But nonetheless, there is ups and downs and tissue specificity of genetic expression. And it would be a little difficult to understand why, for example, all the peripheral tissues would be represented in an organ, that is the thymus, when these T cells develop, and this tissue then has to, or these T cell receptors have to be selected against potential self antigens. Because once they leave the thymus, they go everywhere. They might hit, the most famous example, they might hit the beta cell in the pancreas that make insulin. And if they don't tolerate this, they might destroy the beta cells, and you don't have any insulin, and you know what that causes: a severe metabolic disease, diabetes mellitus. So how is it possible to screen T cell receptors against self-antigens? Now imagine yourself, once again, you are the immunoengineer. You've sort of taken that position for quite often now during these lectures. The simple solution would be to say, well, if I have to do this, I simply expose the T cells and the T cell receptors to all peripheral antigens. Now that, of course, clashes with the classical view that tissue differentiation depends on differential gene expression. So we have a couple of thousand so-called housekeeping genes that are needed by every cell for the normal living of a cell, as it were, and then we have these tissue-specific aspects that distinguish a muscle cell from a nerve cell. But how would it then be possible to represent the peripheral self in the thymus for these T cells to see them? That is a very difficult question. The first answer, the first glimpse of the answer came from studying an inborn disease of autoimmunity that was discovered and described by Finnish doctors. And Finns have the advantage that their population structure is very well known, and there is not much change over time in these different populations. So for human genetics, this is a very useful resource. So is the population in Iceland, for example, but that is only recently coming to the fore. So the Finns, there was a group of people in Finland that suffered from a polyglandular endocrine disorder that was caused by destruction through the immune system of various immune organs. For example, hormone-producing organs. For example, beta cells in the pancreas, the thyroid, where the thyroid hormones are being produced, and many other things. So it was a polyglandular autoimmune disease. And it was found that it could be traced to the mutation of a single gene that is now called autoimmune regulator A-I-R-E, ARI. And this gene was found to mediate, in still I would perhaps, I think probably would go so far to say, in not quite understood fashion, molecular fashion, to destabilize the genome of stromal cells, specifically in the thymus, perhaps also in some peripheral cells, but specifically in the thymus, to cause these cells to express many more genes that they would normally supposed to be expressing, violating the rule of specific, tissue-specific gene expression in the thymus, so that the thymic stroma would express many, many, many genes, and in combinations that would be very unusual for the periphery, in order to make proteins. These proteins would be degraded, we spoke about this. These breakdown products would be loaded onto MHC molecules, and these MHC molecules would be presenting these peptides at the cell surface, so the T cells could go around and scan this complement of MHC peptide complexes, and would also encounter, in the thymus, would encounter peptides that are derived from insulin, for example, and severely autoreactive T cell receptors would be purged from the repertoire. Now, this is, when we look at the T cells, how does that work? One knows from careful in vitro studies and very elegant in vivo studies that we have a situation here, and this is a diagram that summarizes a lot of work. Here is, on the y-axis, we have the % survival of double positive thymocytes. That is to say, double positive thymocytes is an intermediate stage of T cell differentiation, just before they undergo selection. So they express a T cell receptor at the cell surface, and now they have to see antigen. And when they see antigen, they have three options. They see nothing, because the receptor has a structure that is not recognizing either MHC, or it's not recognizing MHC plus peptide. And that receptor is, particularly when it doesn't see MHC, it's not very useful, because if this receptor is in the periphery, when it doesn't see self-MHC, then it's useless. And these cells that have very little affinity to these MHC peptide complexes die by neglect. This is the term that describes this phenomenon. So the developing T cells at the double positive stage requires a signal through the T cell receptor to survive. So the double positive cells that you might know make up most of the T cells in the thymus. 80%, sometimes we could say 90% of the cells are double positive cells. So the thymus is full of developing thymocytes, mostly these immature double positive cells. And they are very sensitive to these survival signals. If they don't get survival signals, they die. They are also very sensitive to corticosteroids. So when you are stressed, the first thing that happens is that your double positive thymocytes in the thymus die. And this was one of the reasons, just in parentheses, why the function of the thymus was obscure for such a long time. Whenever doctors performed autopsies on human patients, they normally did this not on healthy people, except in case of accident, but normally they would do this in diseased people that had gone through the agony of death, and then after death would be autopsied. And during agony, that is in the situation of severest stress, presumably due to some severe disease, for example infection, the thymus would change its structure quite dramatically, because all the double positive cells would be completely wiped out. And we've been discussing yesterday that when they are dying in the thymus, they're immediately removed. So the thymus basically looked like an empty tissue, and pathologists thought, well, what is this all about? A bit of fat cells here, and a bit of epithelium, and a bit of mesenchymal cells. Not very interesting. So the thymus was considered to be a rather useless tissue. One had noticed it's big in infants, but basically non-existent in adults because of this particular situation. So it took a long time before the thymus found its role. And this is the way to do it. These cells have to be very sensitive to survival signals, otherwise we would not be able to get rid of them. So they are dying off when they don't see a peptide MHC complex. They are positively selected, that is, they survive and differentiate further when they make contact. If they make too strong contact, indicating to the T cells, perhaps I'm now recognizing something that is a very good fit, these cells are negatively selected and again die by apoptosis. Now you have to distinguish this phase and this phase. Here the cell dies because it does not see a structure on the antigen presenting cell. Here the cell dies because it has too good a fit. So this is the two extremes. So that must mean that the cell biology of the cell changes dramatically. So here it is dependent on survival signals. Survival signals allow this through the T cell receptor to signal. They are positively selected. If they bind too strongly to the MHC peptide complex, although they have this earlier survival signal, as it were, now this signal is interpreted quite differently. Again, they are being killed. And that leaves the repertoire with a collection of cells that can recognize MHC peptide complexes, but not too strongly. And I'll come to an alternative scenario in a moment. So that means you actually can generate a quite diverse repertoire of T cells by providing them with a single antigen. So experiments that were done by the group of Kapler and Marak, what they have done very cleverly, they have covalently bound a peptide to the MHC, preventing endogenous peptides from binding into the empty groove. So it was already occupied by way of synthesizing this particular molecule. So there was no chance for these protease-generated peptides to bind into a groove because the groove was already full. So the specificity was a monospecificity. Yet the diversity of the T cell repertoire was quite diverse. That immediately suggests to you that it's not a precise sequence that is selected here at this stage. It simply is the capacity of the T cell receptor to bind to MHC, and because MHC is only stabilized by provision of a peptide, that is basically testing for reactivity to self-MHC. And this is what we want, because we want to be able to recognize antigens with our self-MHC. It's no point if my T cells recognize Anya's antigen-presenting cells and not mine, because T cells have to be in my body. So this is positive selection, if you wish, is simply selecting, at least to a first approximation, in particular, the capacity to recognize MHC. And if they then recognize MHC peptide, which normally then increases affinity, then the cells are negatively selected. So here is the model. We have, when the double positive T cell receptor, the double positive T cell, and its T cell receptor recognizes this particular peptide, it then differentiates further. If this interaction is too strong, it will be deleted. Now, in order to achieve this, cells have to be exposed to different environment, because you can imagine it makes sense when a cell has to progress through a differentiation pathway and signals from the outside are interpreted in different fashion. It makes sense to spatially separate these individual steps. And this is just indicated here. Ignore the bottom one here. This is indicating in schematic form and rather abstract form how cells move from various niches in the thymus. So when they exit from the blood vessels, they go through the so-called perivascular space, which is next to the blood vessel. They enter the epithelial space in the thymus. Then they move from one to the next niche, to the next niche, and in this particular niche, by the name of DN3, there is selection for the first rearrangements of the T cell receptor that occur, and then it goes further, like in the scheme I showed you yesterday, until they've reached the final destination. So it makes sense to somehow separate this when you think about the system. And people have been trying to define the individual components that define these so-called niches. Here's one, taken from a recent review by Yosuke Takahama. So what he has defined, that the initial stages and specification of the T cell occur in the so-called cortex, that is in the cortical region of the thymus when you look at it under the microscope. So these early thymocytes progress, and during progression are being exposed to different types of molecules. The details don't really matter, so survival signals are being given, MHC class 1 in that case here, or MHC class 2 providing the peptides. This particular molecule, we'll come to this in a moment, provides signal to specify them to become T cells. So all this seems to be orchestrated somehow, and cells migrate through the thymus, and they often depend on positional sequences, signals provided by chemokines. We've seen chemokines in the previous movies. So all sorts of signals coordinate the developmental progression of developing T cells. And they finally see, for the negative selection, they are exposed to self-antigens, to tissue-specific antigens in the so-called medulla, that is the inside of the thymus. So they are being positively selected most of the time in the cortex, then they move for further testing into the medulla, and in the medulla there are these particular epithelial cells, medullary thymic epithelial cells, that express this autoimmune regulator here, and this autoimmune regulator makes sure, at least in part, that these peripheral antigens are being expressed at the cell surface. The T cell sees them and either binds or does not bind. If it binds, it will be killed. So it's anatomically very well structured. It has evolved over the time, and it ensures that this progression occurs normally. So this is what you read in the textbooks. The trouble is, it also works without this finely tuned structure. And I'll show you some experiments that show you that nature is much more flexible than you might think. It's not always so that when you observe something that this is the prerequisite for the process to occur normally. Sometimes it's simply a byproduct or has some as yet unknown function. So how does this microenvironment that we've now portrayed to be fostering this development develop? I showed this picture many times over now. The thymus is a very ancient tissue, and the amazing situation is that this process that I operationally defined already in, I think yesterday, is probably taking place since about 500 million years. Now, where is the thymic microenvironment coming from? During embryogenesis, it develops from the third pharyngeal pouch. I said that already yesterday. We do not know exactly why that is in the third pharyngeal pouch. There are some developmental signals like wind signals or bone morphogenetic proteins or sonic hedgehog signals that somehow regulate the appearance of a particular gene expression pattern in the third pharyngeal pouch. One part expresses the transcription factor FOXN1 that will feature prominently in a moment. Another part features another transcription factor by the name of GCM2. Names don't really matter. Suffice it to say that the red and the green develop into two different types of organs, the thymus and the parathyroid. So it's precisely orchestrated. The thymus is always in the same place. It originates developmentally from the pharyngeal endoderm. Now, you can transgenically mark, not only in fish but also in mice, this developing tissue. This time we are using this FOXN1 transcription factor because it defines the tissue that later develops in the stroma of the thymus. So this is perhaps a bit difficult to see from behind, but here is a section through an embryo, a mouse embryo at 15.5 days of embryonic development. There is a site of expression for this FOXN1. There is a site of expression for this GCM2. This is the future parathyroid. This is the future thymus. And if we now link to the promoter of this FOXN1 gene a GFP protein, the same story as we heard yesterday. Then we mark these cells with green fluorescence. And we can even make a genetic trick by incorporating into these cells a gene function that makes them susceptible to a particular toxin, and that is in this case diphtheria toxin. So we can inject the mother that carries these embryos with diphtheria toxin. These cells transgenically express the diphtheria toxin receptor. They take up the toxin kills the cell. You see green fluorescence is disappearing. Cells are dead. This is a chemical thymectomy because it eliminates thymopoiesis at later stages. The cells are simply eliminated during embryogenesis, and we can observe the effects of it. This is a system that can be used in basically all organs. The only requirement is that you can define a promoter that drives these particular transgenes, for example, the fluorescent gene or the receptor for the toxin, or you can think of other ways. That then allows you conditionally at a prescribed time in a prescribed tissue to eliminate cells, ablate them, and see what happens. So this is the most precise surgical procedure you can think of. Not even a microsurgeon could do this so precisely. We can do this by these pharmacological means. And that can be used to great effect. For example, if you want to study the function of a particular cell type, you simply target this cell type in this or similar ways and then eliminate it completely. Now, interestingly enough, I showed you this picture before. The thymoid in the lamprey larvae, where these T-cell equivalents of the lamprey develop. It's a bit hard to see perhaps from the back, but we have lymphoepithelial tissue here, epithelial cells encompassing lymphocytes. This is the histological section through a shark thymus. We have a densely populated cortex where all these double positive thymocytes are sitting, and a more loosely populated medulla where most of the selection has already appeared and has already taken place, and there's much fewer cells here. Shark and mouse, densely populated cortex, loosely populated medulla. Nothing has changed over 550 million years. All the more so, it will be surprising, as we will see later, that this zoning of the thymus is not an absolute requirement for T-cell development to occur. It can occur even without this particular zoning process. Now, I'm not discussing the bottom part of the picture. I now come to experiments that we did many years ago when the physiological function of this transcription factor was first revealed. So it was revealed through the observation of a naturally occurring mutant, a mouse mutant. That an equivalent mutant exists in rats, it also exists in humans. It is accompanied by the failure of T-cell development and also by failure of proper hair keratinization. This is why this phenotype was named nude. And you must have seen these rodents, these nude rodents, because they are a great model for transplantation immunology. Because they don't have any T cells, through this problem in the development of the thymus, they can accept any possible organ from any, even from xenogenic origin. So it's been quite popular maybe in the late 70s when I was a student, or mid-70s, when people were transplanting human tumors onto these nude mice. They would accept these tumors, tumors would grow, and they could test then the function of chemotherapeutic agents and see whether these particular tumors from patient X were sensitive to this particular drug, and then perhaps tailor treatment accordingly. But they were also used for a number of other things because they would accept basically any transplant because they didn't have any T cells. And the molecular basis for this particular phenotype was traced to mutations in this transcription factor this is now the official name for it. And when you compare, you can just about see it, when you compare the thorax of a normal mouse at equivalent stages, you see a big white tissue that upon histological analysis gives you this green and red pattern, green indicative of epithelial stroma, red of CD45 positive hematopoietic cells, that is T cells, so they are sitting in the thymus in here and developing there as they should. And when you look at the mutant, you see epithelial cells that don't look quite right because they have cysts in them and don't look differentiated enough, but importantly there is no T cells in this particular tissue, indicating that this epithelium is somewhat defunct in attracting and then modulating T cell response. Now we have to make it a bit darker so we will be able to see this a bit better. Yeah, just for a couple of moments. Yeah, okay. So now you can see that a little better. Green is the stroma, red are the T cells, and in this particular instance, in the mutant, you can perhaps spot a couple of red cells, but only outside, not inside the rudiment. And I showed you the movie yesterday, and I pointed out when you eliminate the function of certain chemokines, the cells come near the thymus but cannot enter. And this is very reminiscent of this. Somehow the cells cannot get in, or maybe the ones that get in don't survive. So that's a very interesting phenotype, and I'll show you what the molecular basis for this is. The molecular basis for this is that this FOXN1 transcription factor, over these hundreds of millions of years, has managed to coordinate the expression of a great number of genes. And I'll just indicate a couple of them and say a few words what they are doing. For example, this transcription factor, it's a lot you can't read now, but never mind. This should be the name of transcription factor. It regulates this particular gene encoding the DLL4 protein. DLL4 protein is perhaps familiar to the students of invertebrate development. It's called delta-like ligand. It's the equivalent of a very famous gene where it was discovered in Drosophila. That is a ligand for another important developmental regulator for Notch. Notch is a transmembrane protein that interacts with its ligand. One of the ligands is delta-like 4. Notch, or rather Notch1 in mammals, is expressed on hematopoietic cells. And when Notch1 is engaging the delta-like 4 ligand, one of 4 ligands for the Notch family, then this interaction stimulates these progenitors to assume a T cell fate. I will show this in a moment in a very dramatic experiment. This FOXN1 transcription factor also regulates the expression of chemokines. By now you can recognize the nomenclature. Chemokine like molecule 25. The exact name doesn't really matter. This is another type of chemokine ligand, number 12. It also regulates the expression of cytokines. Cytokines are molecules that provide survival signals. In this particular case, KIT ligand, the alternative name is stem cell factor. It's a very important hematopoietic growth factor and survival factor. And all of these and many more, which you cannot read now, for somehow they disappeared, are regulated by this one transcription factor. So this transcription factor is basically perhaps some sort of master regulator orchestrating a number of functions that in T cell development in this particular tissue have a particularly important role. Now, When this was discovered, it took a couple of years in comparative analysis across various species over many millions of years of evolutionary time, and I'll give you a summary a little later. It occurred to us that we had to prove that we think we understand the process of T-cell development. Because if you think you understand a system, you have to be able to build it from scratch. Imagine again yourself as an engineer. So you try to do it one by one part, next part, next part, next part, unless you fail, or else you produce the desired outcome. So, and since this one transcription factor has these many targets, and these many targets were selected through a complex SIP selection system based on evolutionary information, as it were, we set out to reconstruct T-cell development. T-cell development by starting from this, a defunct epithelium, and now adding one factor after the other transgenically, instead of bringing back the transcription factor, which of course restores function, but now we wanted to see which factors that this transcription factor regulates are really important for this process. And I'm particularly fond of that experiment. This is why I'm showing it to you. So now this is the logic. It's basically very simple. It took a couple of years though, but it's very simple. So we start with an epithelium that we call is in the dysfunctional ground state. Just doesn't do anything. You've seen this, the cells sitting in the vicinity, but will not recognize this as a thymus where they should go. Remember the video from yesterday where these cells were extremely keen to go to the thymus, as if they knew this is where this was their destination. Here this does not work. The cells do not enter the thymus. Now I've delineated these four factors that I showed you. SCF is the KIT ligand, the stem cell factor. This is this notch ligand. And these two chemokines in this different color code. And now because we are providing these factors individually, we can make all possible combinations. So how many different mouse strains does that give? Quick calculation.
When they do pairwise or?
No, in all possible combinations. Well, more than a dozen.
Yeah.
Okay? So I'm not showing you the phenotype of all of these, but the key phenotypes. So just to give you the idea how that was done by a PhD student, Leslie Calderon. She came from Cuba to my laboratory, and she was keen to do that experiment. And she knew she could work hard, and she had to work hard because she had to micro-dissect these embryos under the microscope, take out these little organs, and calculate or determine the number of cells and their phenotype. So this is the baseline. When she dissects the two lobes of the embryo, of the thymus from the embryo, in total there are about 250 cells. Most of them are myeloid cells, and because she cannot eliminate the capsule during this microsurgery, the cells that are sitting around here are also coming into consideration. So, but the important point is, since these two markers here, CD19 and CD90, you just believe me, CD19 is a marker for B cells, and CD90 in this particular context is a marker for developing T cells. And when you count them, this is about maybe 5 to 10 cells, this is maybe 20 cells. And this is what we consider background, because you can never be sure that you do this right, because there are blood vessels when you do the microsurgery, so there's always some reason for contamination, but very, very few cells. So now she generated these different transgenic mice, took out the thymic lobes, counted the cells, and determined the phenotype. So first interesting phenotype that she observed is when she looked at mice that expressed stem cell factor and chemokine. Chemokine allowing cells to be attracted to the thymus, but now they don't see the normal complement of factors, they only see stem cell factor. And the big surprise was that the thymus was full of mast cells. And mast cells are not normally present in the thymus in large numbers, but in this particular case, the thymus was converted into an environment that was happily supporting survival and differentiation of mast cells. So that is sort of interesting. That already indicates to you there is some sort of modularity in function. Because when all factors come together, we only see T cells. The occasional mast cell, but mostly T cells. And now, in this combination, she has mast cells. Now she adds one more factor to this. That is CXCL12. Mast cells are all gone. B cells develop. And these B cells develop as if they were situated in the bone marrow. No T cells, no mast cells, only B cells. So we can now say CXCL12 brings in a dominant function. It converts the environment into one that does not like and disfavors mast cells. It now favors B cells. Now we're beginning to be in the interesting range because we're interested in lymphocytes. So these three factors, now we've reconstituted a B cell environment. But of course we're not interested in B cells, we're interested in T cells. So we look at this combination. SCF, CCL25 and delta-like 4, but not this one. These three. What we see is the T cells begin to develop, but they arrest at a very early stage. They just cannot go forward. They just come to the thymus and begin T cell development and then stop. And we call them double negative thymocytes because they do not express a combination of markers that would indicate to us that they express the T cell receptor at the cell surface. They don't have T cell receptors, but they are early T cells. And now, of course, this is the combination you would be waiting for. Now all four factors, and all of a sudden with these four factors, it's too much at the bottom, you can't see that now. When you express all four factors together, T cells come, develop, and express the T cell receptor at the cell surface. And that is an amazing result. Because with only these four factors, Leslie could reconstitute the thymopathetic activity for about half of all steps of T cell development, indicating that this is apparently a very robust system that depends only on very few factors. When you rerun the sequence in your mind, then you will see, depending on the combination, you get a particular outcome. And here is now the outcome for all possible combinations, and there is a color code here. So when there is lots of red, it's mostly T cells. When this is lots of blue, mostly T cells, and lots of yellow, mostly mast cells. So it's never a one-to-one thing. But the majority of cells is according to the system that I explained to you. So we can recreate these various situations. And now what is also important, this is now the proportions once again, what is also important is that we can build a certain degree of hierarchy. So we can say one factor dominates the other. I showed you CXCL12 overrides the mast cell program of the two other factors. And when then delta-like 4 comes, B cell development is completely abolished and only T cells can be made. So there is a hierarchy, there's combination, cooperation, synergy, and modularity. Exactly as you would organize it if you were the engineer of a car manufacturer. In fact, all the cars that you see that come from Volkswagen are basically built according to this modular design. They have a different outside appearance, but inside is all the same. So what you are buying when you buy Seat or Volkswagen or Audi or whatever these brands are, it's basically always the same car. It just looks different. So that is a very clever design because it's very simple and it saves a lot of resources. And this is exactly here. So we have a modular design. We have factors that we can use. And you will not be surprised to hear that CXCL12, for example, and stem cell factor are factors that are expressed in the bone marrow, but delta-like 4 is not. So the bone marrow then makes B cells. And this is one way of regulating differentiation in different places. So this is the three key features: synergy, context dependence, and hierarchy. But now I show you the most dramatic result of that series of experiments, which I have not specifically pointed out in the picture before. So this is what I just showed you all 4 factors re-expressed, and we go to the double positive stage. For those of you who have seen a flow cytometric profile of early thymocytes, this is the normal picture. Most of the cells are in this particular quadrant that express both the CD8 and the CD4 molecule. And this is for immunologists the indication that they are midway in their differentiation, express the T-cell receptor, and this is what they look like. Now, what Leslie found was that she could reproduce this outcome. All cells that are being made are double positive by simply expressing only 2 factors. Just 2 factors. CXCL12 and delta-like 4. You see? Same result. A chemokine to attract them, delta-like 4 to tell them you should become a T-cell. So the environment needs only 2 factors to jumpstart a differentiation program, or rather 1 factor, that drives these cells into mid-differentiation. Quite extraordinary. And that is a very important result, as will become obvious in one moment, because when you now compare the gene content in various genomes across the chordate lineage, mouse, medaka is the equivalent of zebrafish, shark, lamprey, this is our envisaged common vertebrate ancestor, and Amphioxus as the basal chordate. What you see, you have a delta-like factor, you have a notch receptor, so the specifying ligand receptor components are there. You have a chemokine receptor here that is already present in lamprey, and you have the corresponding ligand, and we assume the same is true for a common vertebrate ancestor. What you don't have in Amphioxus is you don't have the chemokine and you don't have the chemokine receptor. But what is very important, I showed you yesterday when I showed you the movie in zebrafish that homing to the thymus was dependent on 2 chemokines, if you remember, or 2 chemokine receptors, CCL25 and CXCL12. So this was also very important, because without this, homing was very inefficient. Now this chemokine does not exist in lamprey, nor does its receptor. But the reconstitution experiment now showed us these very early organisms probably did not need this. This was just refinement, creating greater efficiency or maybe more robustness. But in principle, it would work with only these 2 factors. You have to get in the cells, into the environment, then stimulate them, and then off they go. So that's quite a remarkable finding, indicating that this complex differentiation pathway, until they make the T-cell receptor, is regulated by less than a handful of factors. Quite extraordinary, I think. And of course, there's a lot more to do, because when these T cells make their receptor, then they have to be selected. And that, of course, requires, on the counter side, you have to express MHC and make sure that the MHC is loaded with peptides so that the T cell receptors can be tested. So there's, of course, a bit more that needs to be done. But if you were given the task of only generating T cells that make a receptor, then you would be done with these 2 factors. Quite extraordinary. And what wasn't visible on the slide before, that somehow disappeared because of the color, there is another factor that is regulated by this FOXN1 transcription factor, itself a transcription factor by the name of C2TA. And I mentioned this yesterday. When C2TA is mutated, Bear lymphocyte syndrome occurs in humans because C2TA regulates the expression of MHC, or is one major regulator of MHC, particularly class 2. And now we know that FOXN1 also regulates C2TA, and through this cascade then regulates the expression of MHC, which is required not only then to attract, specify, but also then start selecting the cells. And there's another student who works on this problem, has recreated now mice, or has created mice that additionally re-express MHC on the epithelium, and we are waiting for the phenotype to happen. So we can now artificially, although in vivo, but artificially create thymopoietic microenvironments with very few factors Here are two famous ones, and they probably recreate the first type of epithelium without its coordinated microstructure, but that is important for the T cells to develop here. And we can also think of creating tissue artificially by using the now very popular procedure of stem cell manipulation. So you know that you can induce from any somatic cell more or less pluripotent stem cells, and you can find ways, or people are working on this, you can find ways to differentiate these cells into the thymic epithelium in order to then artificially build the microenvironment that would foster T cell development. So we have a break now, and after the break I will be discussing what we have learned about the alternative function of MHC. If we go just back for one more, no, it's not the right picture here. MHC presenting the peptides for the selection process, but why MHC and MHC diversity is also important for the species or the population structure as a whole.